Hello, everyone, and welcome to our online lecture series for plant biology. Today, we finally get to our land plants in our discussion of plant evolution and diversity. You'll notice in a lot of these lectures, I like to recap things that we've talked about previously, so that's where we're going to begin. So last time we talked about how life evolved on planet Earth in the first place, coming from an aquatic, anoxic environment, evolving into simple prokaryotes like bacteria, and the slightly more complex archaebacteria with their cytoskeletons that live in places reminiscent of our old Earth. Now, one subset of this group became much larger with membrane-bound organelles, these being the eukaryotes. Now, all eukaryotes inherited our endosymbiotic event with the alpha proteobacteria that became mitochondria. They diversified into many different lineages today that we don't spend a lot of time talking about any of the other ones other than the archaeplastids. Because in the archaeplastids, or the old plastids, we had another endosymbiotic event, the engulfing of a cyanobacteria that was photosynthetic. And all of the descendants, from the red algae to the different types of green algae, to today, some of our early land plants, the liverworts, mosses, and hornworts, that we collectively call the bryophytes, all inherit this singular endosymbiosis of cyanobacteria. But we know that this endosymbiosis wasn't the only thing that defined this group. We know a little bit about their life cycle. For example, they were haploid dominant and inherited the chlorophyll A pigment from their cyanobacteria. Now, as these things evolved into symbiotic organisms that today we call chloroplasts, we know that that gave them some opportunity for differentiation. We see our first major differentiation event between the red algae and what would go on to become the green algae. So this branch point right here. And of course, with each branch point, we have traits, synapomorphies, that are specific to each group. The red algae, as hopefully you'll remember, are differentiated from the green algae by a couple of different mechanisms. Two in particular that have to do with the way that their chloroplasts function. We talked about the presence of phycobilin proteins, those being the red pigments that give algae its red name, as well as the fact that their chloroplasts have no grana. Instead of thylakoids being arranged in stacks, they're equally spaced apart. And they also have a slightly different variant in terms of their starch storage. And then, in my opinion, probably one of the most interesting things about the red algae is the way in which they evolved their multicellularity. Rather than the plasmodesmata that we'll see later on, red algae use the pit plug and the pit plug for the pit connection in order to establish joining elements between cells so that way bodies can be multicellular. Now, looking down the branch that became our green algae, we know that they evolved their own different types of features that separate them from the red algae. Most notably, we talked about the different starch variant that they have. We talked about the fact that they have a new chlorophyll pigment, a chlorophyll B. And we mentioned that a large number of our green algae developed a phycoplast type development of cell division. Now, last time we didn't focus on it, but your book does mention it, so I've included it here. There's also a particular type of flagella in terms of its structure, called stellate flagella, meaning star-shaped in its molecular structure, that all of our green algae inherit. And these characteristics, particularly the dominance of the phycoplast, is what we see preferentially in a lot of our chlorophytes. But we know that not all of them have that parallel microtubule structure to the cell plate. If you need a refresher on that, please make sure you go back and understand the difference between the cell plate and the phycoplast being the microtubule organelle and the cell plate being the actual building of the cell wall. And there is some variation there. That's why this feature can kind of pop over. It's not universal. But what is universal is what separates all of our chlorophytes from our charophytes. That being the presence of a phragmoplast rather than a phycoplast, where the microtubule elements are perpendicular to the building of the cell wall. We also talked about the fact that our charophytes are multicellular via plasmodesmata rather than pit plugs or any of the other multicellular mechanisms that exist in some of our independently evolved multicellular algae. Then we also talked about the fact that their cellulose construction machinery, which last lecture looking back at, I realized I misspelled cellulose constructure, not construction. Sorry about that. Is in a rosette octamer. So again, a kind of rose-shaped structure that helps build the cellulose fibers for the cell wall of charophytes. And then your book also emphasizes that they have filamentous apical growth, meaning they'll oftentimes have multicellularity that leads them to be more string-like, and they will always grow at the tip. Now, this does not mean that they have a true apical meristem just yet. We have to wait a bit for that to be the case, but we see a trend towards this in our charophytes. And so today, we'll explore the different features and the different species that make up our first land plants. This group here, collectively known as the bryophytes. So bryophytes are thought to have separated and arrived on land about 450 million years ago. And from our discussion last time, we know that one of the major traits that separate our land plants from our charophytes was that specific form of their alternation of generations, wherein bryophytes go through the system that you are familiar with, where mitosis is performed in sporophyte and gametophyte stages, and meiosis and fertilization punctuate the separation between the two. Now, one of the things that we see consistent in our alternation of generations that is separating land plants from their green algae ancestors is a multicellular sporophyte, meaning that the diploid stage is performing mitosis. So to really emphasize where that is, we've included it here as one of the major inherited traits. They also, in their alternation of generations, will create specific organs that are able to perform meiosis and are able to create sperm and egg. The organs that create sperm and egg are our gametangia, and it's usually an either-or situation, or can be an either-or or a both, depending. Some bryophytes have gender, some bryophytes do not, so we can't make a blanket statement on that. And then the sporangium are the things that make the spores. And what else is important about these structures is that they're enclosed. They are now protected from the environment. This is very important in helping these cells survive in a dry environment. The next couple innovations we will list actually have to do with protecting our land plants from drying out. Notably, the bryophytes develop a cuticle, a waxy layer on the outside of their cells. Now, everything that we learned about the apical meristem and the three different types of plant tissues, the ground, epidermal, and vascular tissues, does not apply to bryophytes. They only, for the most part, have one tissue type that has slight differentiations. So their internal cells have the same genetic imprint as their external cells for the most part, but their external cells will make a cuticle. And in the cell layer that is going on to make the spores being the things that will travel around and make a new gametophyte, those spores are lined with a brand new sugar polymer in order to help prevent them from drying out. That polymer is called sporopollenin. Sporopollenin is one of the most inert chemical substances on our planet, meaning that it's not going to be reacting with anything. It provides a protective layer for the outside of spores and is still utilized today in pollen. You can see the very particular structural patterns that sporopollenin can play a role in creating in this scanning electron micrograph of different pollen grains. Now, the inertness of this sporopollenin actually allows pollen grains to survive for millions of years. In this way, we're actually able to tell what plants were around in what abundances in things like lake sediments, because we can analyze what pollen grains are there. So not only is this important for purposes of plant biology, but sporopollenin also gives us insight into the past into studying our own environment. Kind of cool. Additionally, another protective layer that we see in these structures that are making our egg and sperm is a sterile jacket. Sterile jacket for our antheridia and archegonia. Antheridia being the specific structure that makes sperm, comparable to an anther of a flower, and archegonia being the structure that makes eggs, comparable to the ovaries of a flower. But remember, these are not flowering organisms. They do not make the same reproductive structures. They do not make seeds. These are spore reproducing. And so, you can see that they inherit that type of reproduction ancestrally. And it will be a while before we switch from spores to seeds. It's a couple lectures down the road. But one of the important steps in changing these spores to adapt to land is the retention of the zygote, the newly fertilized egg. Rather than the egg being fertilized somewhere out in the environment, in our land plants that are now migrating into drier environments, the zygote stays with the mother plant. This allows the mother to create a very favorable environment, like a more wet environment, for the spore to have, for the new zygote rather, to have a chance to succeed. The zygote will then go ahead and make its sporophyte attached to the mother gametophyte, and from there, the spores will be produced in our sporangium and set forth to explore the world and continue to colonize new places. We will have a closer diagram of this process later on in the lecture. So hopefully you can start to recognize some of the terminology I use in my explanations, and by the end of it, you're able to follow along like a complete story. And then our last major shift in our bryophytes is that they have a true apical meristem. The type of growth that we are seeing is now reminiscent of the type of growth that we see in all land plants. Having a region of cell division, having some region of elongation, and having some, though very little, maturation. Keep in mind, again, that before you can have 3 different types of tissues, you have to have started with 1. And our bryophytes are the group that has essentially 1 tissue type. No separate vasculature, no truly separate epidermis. It's mostly just the ground tissue. So with those evolutionary innovations, I also like to always include some basic descriptors of the group. Things that you'll see commonly stated, whether it be in the textbook or on Wikipedia pages or in other plant-focused classes. We know bryophytes as the non-vascular plants. This means that they have no conductive tissues. They do not have specialized xylem and phloem. They therefore do not get tall. Without really effective ways of moving water, you're limited by forces of diffusion through ground tissue as to how tall you can get. One of the things that's also associated with this lack of vascular structure is an absence of lignin. Lignin is one of those important polymers that helps give structure and conductibility to our vascular cells. So, they don't have it, they don't have true xylem, they stay short. Bryophytes are also oftentimes referred to as a paraphyletic group. And I will say that there is some contention about this, but generally the way that I've been taught it thus far, and up until evidence is presented that's convincing enough to the contrary, we will represent the relationships between liverworts, mosses, and hornworts as a staggered staircase, with liverworts peeling off first, mosses peeling off second, and hornworts peeling off third. Now, because we cannot trace these 3 groups to a common ancestor that does not include all land plants, because that's what's going to be here, further vascular land plants, because these don't group by themselves without including our land plants, we call them paraphyletic. If they were instead connected to one node on their own and vascular land plants were elsewhere, they would be considered monophyletic. But because this group needs to contain not only the liverworts, mosses, and hornworts, as well as the land plants, the vascular land plants, in order to contain all descendants, bryophytes are referred to as a paraphyletic clade. For the most part, these bryophytes are mostly tethered to wet environments. So you will see them dominating in places that are very moist constantly. But they do have some variation in that. You will occasionally find some bryophytes in drier areas, but they'll wait to reproduce up until it becomes wet. They are also gametophyte dominant. That is where the vast majority of their body is actually descended from our haploid cells. So when we talk about the green photosynthetic tissue of our liverworts, mosses, and hornworts, that's actually the body created by the haploid cells. These haploid cells have the equivalent genetic material as egg and sperm. So while they are not technically egg or sperm, the gametophyte will produce the egg and sperm, they have the same genetics as them. So I will oversimplify things frequently and refer to them as the sperm and egg bodies. And it's really weird to think about it when you understand humans. Sperm or eggs in animals are a punctuated cell line, meaning that they have just one cell that has to fuse or die, but the sperm and egg of plants can go on to be photosynthetic, set up its own shop, gather its own resources, and live on. So that's a very, very unique feature and inherited ancestrally. We also will see in all of our bryophytes an unbranched sporophyte, meaning that the sporophyte has very simple growth. It will start from the zygote, the fused diploid egg and sperm, and it will grow from that zygote till it creates sporangia. And from there, you'll repeat the cycle of the alternation of generations. The spores will be generated via meiosis, and the process of generating a brand new gametophyte body will begin. The fact that their sporophytes are unbranched means that their sporophytes still are fairly simple. And we know that this will change quite drastically when we get to talking about our vascular plants. All of our flowering plants, all of our cone-bearing plants, well, they've got highly branched sporophyte bodies that are now diploid dominant, shifting away from our haploid dominant past. So let's take a look at some of these different organisms, some diversity. Here on the left, we have drawings of various different liverworts, an actual picture of some of our liverworts, a picture of a moss with the gametophyte base that's photosynthetic, and the nutritionally dependent sporophyte that's unbranched, sticking out. And then we have hornworts. These look similar to, or can look similar to our liverworts because they have similar body plans. Now, for some vocabulary in terms of anatomy, I'll first introduce you to the term thallus, or thalli. The thallus refers to the undifferentiated body of a gametophyte. Like what we're looking at in our hornwort here and our liverwort here. These thalluses, or thalli rather, sorry, thalli is the plural on it, are free-living and photosynthetic. And this is that egg and sperm body that I was talking about. While they are not technically those exact cells, they're not genetically different from them, and they will go on to make them. Now, these thalli can occasionally have rhizoids. Not true roots, but anchoring structures. They do not perform a lot of absorption, because absorption will occur both in the upper tissues and anywhere that the plant contacts the resources it needs. So because you don't have specialized structure for absorption from the ground, the entire body is able to perform absorption of nutrients from whatever it's growing on top of. There's not a lot of separation between these areas. Remember, bryophytes are very short, and it's much easier to diffuse nutrients over small distances, and therefore they don't need that specialization of roots. Now, we'll also refer to gemma or gemma cups. These are cells that can make new individuals via asexual reproduction. You actually see some gemma cups present in this liverwort here, the little circle-shaped things. So what happens here is that there are cells in the cup that are ready to go on to make more gametophyte. So it's gametophyte coming from gametophyte, or asexual reproduction. But they're very clever in that the gametophyte itself needs a lot of water in order to grow. These gemma cups have these very particular wall structures that require raindrops in order to splash on the cup to spread the cells around so that way new individuals can grow. Now, if you're waiting for rain to spread your cells around, you're waiting for the ideal conditions in order to continue growing your liverwort gametophyte. So gemma cups are both environmentally adapted as well as a unique method of reproduction that we'll see replicated in different ways across different plants. Now, oftentimes, all of our liverworts, mosses, and hornworts are considered epiphytes, meaning a plant that grows on top of things. Oftentimes, that plant can grow on top of other plants or things like rocks, where soil is not. They do not have, again, root structures. They have simple rhizoids. Now, to put all of those innovations in one place, we'll again address some of the innovations that we see in our bryophytes. Of course, sporopollenin is one of the major ones in terms of helping prevent our spores from drying out. But we also have, and you see sporopollenin here in the X sign, or the outside of the spore. But we also have structures that can help these spores move around. They're attached to the surface, but then will change shape and be able to fling the spores far away from where they first grew. These structures are called elaters. And the way that elaters work is that they are basically sugar molecules that have a lot of tension in them. So they're these large sugar polymers that when they're wet, they have tension, and that tension will snap the structure when it dries out. And that quick flinging, that quick motion of snapping when it dries out will help push or propel the spore away from the parent. You might be familiar with some analogous structures in some of our seed-bearing plants, like everyone's favorite helicopter seeds, the samaras of maples, where the wings on that maple seed help it twirl far away from where the parent was. They're able to disperse the seeds farther away. In the same way, elaters are helping disperse the spores of our bryophytes. Now, these spores are also fed from a very particular material, similar to what you might consider a nutritive placenta. We call that sporogenous tissue. And this idea of actually providing nutrients to the next generation is one of these major innovations. Historically, in green algae, everything kind of operates, you get the cell, and then the cell has to go on and gather its own resources. We start seeing the investment into future generations, whether it be the haploid spore or in the future the diploid zygotes that will become our seeds. We see investment in future generations as a major evolutionary innovation that helps plant species diversify, adapt, and thrive on our planet. So it's important to note and keep track of how these things change. Bryophytes actually feed their spores through sporogenous tissue. They give them a starting point, at least a backpack full of food to begin with. Now, alongside elaters, I forgot to mention this, we also have other structures. Not all spores in bryophytes have elaters. Some of them instead rely on a structure called an operculum, which will function similarly in its purpose, at least, to elaters, in that it's a dispersal mechanism that again operates on drying out. When these sugar polymers dry out, they snap, and in snapping, they will then release a huge amount of spores a lot of different directions. And while we don't have true vascular tissue in our bryophytes, what we do have is some slightly specialized cells that are able to transport larger amounts of water that we call hydroids, and larger amounts of sugar, leptoids. Now, these are not vascular tissues. They are these parenchymal tissues, but they do have some differentiation and some different role than their neighbors around them. In fact, some lineages of mosses have even, in parallel, evolved hydroids that are dead at functional maturity. Tell me if that sounds at all familiar to our xylem. Cells that hollow out the plasma membrane in order to better transport water. It's pretty cool that we see some degree of convergence between these important vascular structures. I will point out that this is similar to different lineages of what would one day become animals evolving circulatory systems separately. We do actually see this in the different circulatory systems between organisms like you and I and insects, closed and open circulatory systems. So it's not unheard of, but it is cool to see how different lineages innovate and adapt given the machinery that they have. So hydroids and leptoids are pretty cool. Now, why do we call liverworts liverworts? Well, it was once and for a long time believed that liverworts were good to eat if you had diseases of the liver. Now, this is quite simple to explain. A lot of old modes of medicine thought that if you ate something shaped like the organ that was diseased, that shape inherently had to be helpful or restorative to that organ. So liverworts, wart meaning plant in Old English, not at all the wart that you assume it relates to today. Now, liverworts, because of their shape, were oftentimes prescribed to be eaten if someone was thought to be suffering from a disease of the liver. Thank goodness medicine has improved over time. We also have a couple of cool facts about our mosses. So here we have two different mosses. The first is sphagnum, maybe better known as peat moss. It's an incredibly diverse lineage of mosses. There are something like 300 different species of sphagnum on planet Earth, 380 approximately. So quite a large number of species for one single genus, but they're very notable because something like 1 to 3% of Earth's surface is occupied by environments that are shaped by peat moss, things that we call peat bogs. Peat bogs are largely found in cold environments, like the tundras, and occupy a very important ecological niche for our planet. These peat bogs are largely acidic. Because of the accumulation of different minerals and resources there. And they are largely made up of these sphagnum mosses that are occupying the vast majority of the photosynthetic space. Now, these peat bogs actually create environments for other really, really neat plants. Peat bogs are also occupied by different ericaceous shrubs, or the heather shrubs, the blueberry shrubs. I'll put that in quotes just because it's not true that they're all blueberry, but they're in the same family. And a lot of carnivorous plants. Like around here, if you go hiking in a bog, that's where you're most likely to encounter our West Michigan native carnivorous plants. Peat does perform a very important economic role. It was historically used to generate a lot of heat within households, and we still use peat moss as soil amendments in a lot of agriculture. The material is very commonly found and used throughout human history, as these peat bogs were oftentimes anaerobic locations, places without oxygen. So whenever humans, for example, would die in a peat bog, these anaerobic conditions would preserve them very well. So we have a lot of clues about our own past and what uses we had for peat, including food preservation, as we found 2,000-year-old butter in a peat bog, which is really interesting. And so you'd have multiple economic uses of peat both today and in the past. Now, our other image of a moss here is that of Dawsonia. The largest, tallest of the mosses at something like 50 centimeters at its greatest. So that's fairly tall. 50 centimeters being approximately 20 inches. So over 1.5 foot tall moss. That's pretty incredible for something that does not have true xylem, but does have its own kind of convergent type of cells that can help transport water, even if they're not different types of tissues. So next up in our discussion is going to be to look over the process of alternation of generations in our mosses. This is super important, and while I have this image preloaded from Wikipedia, as their drawing abilities are going to be far better than mine, I do hope that you try to copy this down in your own writing to really understand what's happening. But before you try to copy it out, let's narrate the story. You always should be able to narrate a story to an image. So we'll begin with our haploid spores. These have 1n amount of genetic material, meaning that they have the amount of genetic material needed to become sperm or eggs. But spores, rather than immediately becoming sperm or eggs, will be able to perform mitosis. And in performing mitosis, they develop into gametophytes. And in this case, we have a gametophyte that's separate for the male and a gametophyte that's separate for the female, both being haploid. They don't really look different, right? They're both just green bodies, but the end structure that they're making will indeed be different. Male gametophytes will create antheridiums, or antheridia, at their tips, and female gametophytes will create archegonia at their tips. More on what these structures look like in a bit. Now, the antheridia will release sperm that then swim over, oftentimes via droplets, to encounter an archegonium and fertilize the egg. Once the egg is fertilized, it creates our zygote embryo. And this is what I referred to when I said earlier, retention of the zygote. The embryo stays in the body of the gametophyte. So that way you will see the sporophyte, the entire body that's diploid, physically attached and on top of the gametophyte. The sporophyte will create a spore-producing structure, the sporangium, wherein meiosis will occur. So you have meiosis in the sporangium, and that meiosis will create lots and lots of spores that in this case are released via the presence of an operculum that dries out. So please take a moment here to draw this process out for yourself. It's critical in our understanding of plant evolution. We will be looking at the alternation of generation cycle, or at least discussing it, for every one of our major plant lineages. The shifts are very important. You have the gametophyte as our photosynthetic body in bryophytes. That won't always be the case. When we look later on in the plant evolutionary tree, that has switched over to being the sporophyte as the photosynthetic body, with the gametophyte being nutritionally dependent on the sporophyte. For now, however, the sporophyte is dependent on the gametophyte. Looking at this specifically in a typical moss, we will additionally see some of the other vocabulary terms that you may encounter in the labs in this class. We of course know that our sperm are created at antheridia, sometimes all conglomerated together at an antheridial head, and that our eggs are made in an archegonia. The egg carrying structure has an outer cell layer here called the venter. The venter will have a hole in the top called the neck canal, and it is down the neck canal that the sperm can swim. The sperm coming from our antheridia. Once the sperm has swam in, it will fertilize the egg, making a zygote. The zygote, growing and growing and growing, getting nutrition from the gametophyte. It will then grow what's called a foot connection, a setae stalk, and a capsule. The capsule being the structure that's going to go ahead and store and create our future spores. So you can always remember, even though I understand the naming on this is confusing, but the sporophyte is named because it makes spores. Spores are diploid, meaning that the sporophyte, spores are haploid, meaning that the sporophyte is diploid, and the gametophyte makes gametes. Or our egg and sperm. In the sporophyte, you are performing meiosis to make your spores. And in the gametophyte, you are undoing the process when your egg and sperm fertilize to make our zygote. I promise, even though this seems kind of confusing, you will get there. So again, antheridia makes sperm, archegonia makes eggs, eggs are retained, the venter is the layer on the outside that provides the home for our zygote and embryo. Once fertilized, the embryo will grow into a sporophyte that has an attachment at the foot, a setae stalk, and a capsule that's going to hold all of your spores. The capsule can have an operculum or a top cap that is our drying out feature that will help spread the spores. An additional covering called a calyptra. Not going to be a vocab word for our class, but operculum will be, as it is a drying out structure that helps spread spores. The spores themselves, myospores created from meiosis, will go ahead and germinate after release, start creating rhizoids to anchor themselves down, and then form a protonema, or the early body of our moss. Now, this moss can grow in punctuated rhizoids. These gametophytes are our non-vascular quote-unquote leaves with a base of a brown stem at the bottom. And then at the tips of our gametophytes, if they are of different gender, you'll have some that make antheridia and some that make archegonia, and the process repeats, with mitosis occurring here in making our gametophyte body, and mitosis occurring here in making our early embryo into the sporophyte. That can be a lot indeed. Let's preempt some of the things that we're going to look at under the microscope by taking a look at our different tissues, an antheridia, and an archegonia, under the microscope. So some of the features that you'll notice on our Antheridia, and these two are structures from Marchantia, the liverwort. Our Marchantia antheridia has a stalk at the base. It has the tissue that's going to make our sperm, called the spermatogenous tissue. It's going to feed the sperm as they develop. And then importantly, they have that sterile jacket layer, a layer that separates the sperm from the rest of the gametophyte, an interface between the two. In parallel, in our archegonia, our three developing archegonia here for our Marchantia, and our more developed, fully developed archegonia here on the right side, we again have a stalk to separate it from the body of the gametophyte. We have the venter cells that create the enclosing capsule for our egg. And then associated with that egg and venter, we have the neck canal cells that allow sperm to swim down them. So there we have it. A closer look at the reproductive structures, the Antheridia and Archegonia. More to come in lab. A detailed look at the typical life cycle of moss. I know it can be a lot, but I promise, draw it out a couple times, and it will make sense. A look at the overall trends in alternation of generations. Some examples and terminology of common important moss terms. And then, of course, looking back at probably the most important component of our lectures, the evolutionary innovations. So we'll go ahead and end our lecture by talking about some of the unique features and tick marks that make bryophytes different from all of our other plants, and one of the trends that will lead into our vascular land plants. So, liverworts have no stomata. They have no ability to control whether their holes on their leaves are open. Therefore, we call them pores. Without regulated guard cells, they're not stomata. We see the first stomata inherited by mosses and hornworts. So liverworts are our only bryophytes that lack stomata. Stomata unify our mosses and hornworts, and because they are similar in structure of guard cell, it's easier phylogenetically to assume the parsimonious conclusion that stomata evolved once and were inherited by mosses, hornworts, and vascular plants, rather than evolving separately in these different lineages. 3 tick marks versus 1. Now, mosses and hornworts also are unique in the fact that they have air spaces in their leaves to exchange CO2. They have our first spongy space. I won't call it mesophyll because we're not in the middle of a leaf, but it is analogous to the mesophyll, an area where you can exchange CO2 and oxygen within the photosynthetic structure of the plant. Now, mosses differ from our hornworts in that the sporophyte of our hornworts live throughout their entire lives. They don't just produce the sporangium and then be done. They will stay alive. They have a persistent sporophyte. Now, of course, we know this to be the case in our vascular plants. The tree that you lean on is a sporophyte. It is persistent. It continues to grow. The gametophyte is more temporary. Mosses, sporophytes will end after the spores are produced, same with liverworts. In our hornworts, they have persistent sporophytes that are assumed to be shared with vascular land plants. However, liverworts, mosses, and hornworts all have unbranched sporophytes. We see the branched sporophyte being the trend leading to our vascular land plants. There's one additional little feature that makes hornworts unique from the rest of our liverworts and mosses, but more in common with some of our early green algae, is that liverworts and mosses have lots of chloroplasts per cell, whereas hornworts will generally have one giant chloroplast. Other organisms that had one giant chloroplast were things like Chlamydomonas. So that's unique, interesting, an example of potentially convergent evolution, but we can't exactly rule out that the liverworts and mosses, had this ancestral version of one chloroplast per cell, but then independently evolved multiple chloroplasts per cell. We don't know, and we don't have the evidence to say one way or the other, but we do know that hornworts today have a single chloroplast. So that'll do it for this lecture on our bryophytes. Please keep track of these evolutionary changes, as we will very quickly shift from talking about all of our different Archegonia and Antheridia and the gametophyte dominant generation, and instead focus on how plants shifted to sporophytes with differentiated tissue layers.